The biological sex of a human being is determined by the chromosomes that make up that baby's genetic material. Females usually have two X chromosomes, while males tend to have one and one Y chromosome. Research suggests that the Y chromosome has been rapidly degenerating over the course of human evolution, with the gene possibly disappearing altogether. The Y chromosome has seen significant changes over the past 166 million years, losing most of its 1,600-odd genes at a rate of nearly 10 per million years. The X chromosome contains approximately 900 genes with multiple functions, while the Y has approximately 55 genes, with only 27 of them being male-specific. Many of these genes are present in multiple copies, most of which are inactive, lying in giant loops of DNA. Most of the Y is made of repetitive junk DNA. With such an unstable composition, the Y chromosome is at risk of completely disappearing over the course of multiple generations. The early proto-Y chromosome was originally the same size as the X chromosome and contained all the same genes. However, because males only have one copy of the chromosome, it does not have the opportunity to go through genetic recombination, which helps eliminate damaging gene mutations. Without recombination, Y chromosomal genes degenerate over time. If the Y disappears, humans might become extinct or evolve a new sex gene that defines new sex chromosomes. This type of evolution will take millions of years. Some experts believe that the Y chromosome will not disappear at all, claiming that it has several defense mechanisms in place. Scientists have created mice with two biological fathers by generating eggs from male cells, a development that opens up radical new possibilities for reproduction. This could ultimately pave the way for treatments for severe forms of infertility and raise the prospect of same-sex couples having a biological child together in the future. Katsuhiko Hayashi, who led the work at Kyushu University in Japan and is internationally renowned as a pioneer in the field of lab-grown eggs and sperm, predicts that it will be technically possible to create a viable human egg from a male skin cell within a decade. Previously, scientists have created mice that technically had two biological fathers through a chain of elaborate steps, including genetic engineering. However, this is the first time viable eggs have been cultivated from male cells and marks a significant advance. Hayashi's team is now attempting to replicate this achievement with human cells, although there would be significant hurdles for the use of lab-grown eggs for clinical purposes, including establishing their safety. The technique could also be applied to treat severe forms of infertility, including women with Turner's syndrome, in whom one copy of the X chromosome is missing or partly missing. Professor George Daly, the dean of Harvard Medical School, described the work as fascinating, but added that other research had indicated that creating lab-grown gametes from human cells was more challenging than from mouse cells. The study relied on a sequence of intricate steps to transform a skin cell carrying the male XY chromosome combination into an egg, with the female XX version. The cells were reprogrammed into a stem cell-like state to create so-called induced pluripotent stem, iPS, cells. The Y chromosome of these cells was then deleted and replaced by an X chromosome borrowed from another cell to produce iPS cells with two identical X chromosomes. Finally, the cells were cultivated in an ovary organoid, a culture system designed to replicate the conditions inside a mouse ovary. When the eggs were fertilized with normal sperm, the scientists obtained about 600 embryos, which were implanted into surrogate mice, resulting in the birth of 7 mouse pups. The baby mice appeared healthy, had a normal lifespan, and went on to have offspring as adults. Professor Amander Clark, who works on lab-grown gametes at the University of California, Los Angeles, said that translating the work into human cells would be a huge leap because scientists are yet to create lab-grown human eggs from female cells. A brown bovine in Brazil has become the first transgenic cow to produce human insulin in her milk. The mammary gland, designed by Mother Nature, is designed to efficiently make protein, which could help hundreds of millions of people worldwide. The team, including scientists from the University of Sao Paulo, developed the insulin-making cow through genetic engineering. To produce the transgenic cow, a segment of human DNA was inserted into the cell nuclei of 10 cow embryos, containing coding for proinsulin, a precursor of the active form of insulin. Genetic engineering targeted the human DNA for expression in breast tissue only. The altered embryos were implanted in the uteri of normal cows in Brazil, and one transgenic calf was born. After artificial insemination failed, hormones were used to stimulate her to produce milk for the first time. The milk contained detectable levels of human proinsulin and insulin itself, which researchers didn't expect. However, the insulin and proinsulin would have to be extracted and purified for use, and each liter of milk contained only a few grams. Researchers are hopeful that they will achieve greater success with pregnancy and full lactation cycles in the next generation of animals. Eventually, the goal is to create transgenic bulls, mate them with transgenic females, and create a purpose-built herd. Even a small herd could quickly outpace existing methods for insulin production without needing highly technical facilities or infrastructure. A 100-head herd, equivalent to a small Illinois or Wisconsin dairy, could produce all the insulin needed for the country.
Welcome to my channel. Like and subscribe.